What if I told you that your body has a library of every pathogen that has attacked you, as well as a library of the complementary antibody that neutralize that pathogen? And what if I told you that we as scientists had a way to tap in and utilize this library such that we could revolutionize medical diagnostics as well as disease therapeutics? My name is Rafael Smith, and I'm here today to talk to you about antibody specificity repertoires. In the fall, I'll begin my second year at UCSB as a biochemist, and this past summer, I've been working in the Doherty lab with Joel Bozakowski, and we've been studying antibody repertoires. Now, what exactly is an antibody repertoire? Well, every one of you has a unique pool of antibodies that change and evolve based on the immune events that occur specific to your life. And you can kind of think of antibody repertoires as a unique fingerprint. And there will be a lot of overlap. Just like many of us have dealt with the common cold, flu, chickenpox, you will find antibodies for that in your repertoire. But then some of us have more specific isolated incidents, such as when I suffered from Streptococcus bacter, or a bacteria that comes from chicken. So my antibody repertoire will have antibodies specific to this bacterial pathogen that yours will not. And it's this kind of hypothesis that rests our research goal, in that we want to compare serums of healthy patients with serums of diseased patients. And we want to look and isolate disease-specific antibodies. And we can use these antibodies as potential biomarkers. Biomarkers that can be used to diagnose the disease or even treat the disease. Now, before we go into the process of isolating these specific antibodies, a little information about the antibodies themselves. They bind to what's called an antigen. And an antigen is just basically the substance that induces the immune response. If you were to zoom in a little bit, you would see a peptide. Peptide is just a chain of amino acids, and the length of which is irrelevant. If we were to hone in on this a little bit more, these 4 amino acids are supposed to represent an epitope, or the precise specific region that causes the antibody to bind to whatever this is. So these 4 amino acids are responsible for this antibody binding interaction. So really quick, antigen causes the immune response, peptide is the chain of amino acids, and epitope is really this key term because what we're looking for is the genetic information within the epitope, and something about that information is disease-specific that will help us lead to diagnostic tools and eventually therapeutics. And now the disease that we're targeting is AMD, or age macular degeneration. And AMD attacks your eyes, and what it does is in the back, where all the photoreceptors are in your macula, it basically damages it. And it does this in one of 2 ways. Either the blood vessels in your eye will swell, burst, and cause scar tissue to come back, which you cannot see past. Or there's dry AMD, which is the aggregation of this stuff called drusen in that same location in the back of your macula. And so this is what it's like to see normally every day. And this is what it's like to see with AMD. And so you can imagine how difficult it is just to recognize faces, just talking to people or even driving. So there's definitely some great cause or need for some biomarkers for this disease. So our research goal as a whole, what we want to do is identify a disease-specific peptide and eventually determine what the complementary antigen is, or the entire protein is responsible from this peptide. And we're going to accomplish that through a process called bacterial display. And for much of this presentation and much of our research, we focus more on how to identify something that is disease-specific. And the complementary antigen, going from step 1 to step 2, is something that will be worked on later on. So, bacterial display as an overview. The idea is to manipulate bacteria such that they will display peptides, and then we can screen antibodies against these bacteria and figure out which antibodies are binding to which peptides. And it all hinges on the idea that we have this giant library of peptides. So we have 7.6 billion unique peptides, each of which are 12 amino acids in length. And we're engineering them into a culture of bacteria. And so these bacteria will then be displaying each a unique peptide, and we can then add antibodies, much like this, and we can isolate antibody-peptide interactions that do bind, and this is what we want to sequence. So we want to sequence this DNA because something about the genetic information within this peptide is causing this patient's antibodies to bind. And that's a potential candidate for something that could be disease-specific. So slightly more in-depth to the bacterial display process is we're thinking about how we want these antibodies and we want to figure out what peptides they're binding to. So we cannot allow them to bind to E. coli or the bacterial vector that we're using naturally, because that would not be something that would be disease specific. That would be specific to E. coli, and we're not focusing on E. coli. So we initially mix serum with E. coli in order to isolate these interactions and get rid of them, because we only want the antibodies that will not bind to E. coli. So we spin them down, the cells go down in a pellet, we extract the supernatant, and we keep this now newly depleted serum. We can then take this serum and combine it with a new culture. This culture is slightly different in that it's no longer simply E. coli, it's now displaying a peptide. And if we were to zoom in, it would look kind of like this, where we have the outer membrane, this protein, or an ECPX protein that is on the transmembrane of the bacteria, and then engineered right on the end of it, on the N terminus, is the peptide. And this will be permuted all around the transmembrane, or the cell wall, or the transmembrane, excuse me. So the first binding event is going to be antibodies plus bacteria. We allow those to bind, and then we're going to add in magnetic beads. And the magnetic beads is how we can then isolate and extract these antibody-peptide interactions. And what it looks like is there's two possibilities. We will either have a bacterial cell with an antibody and then attached to a magnetic bead, or a magnetic bead with an antibody and an antibody that did not bind to a cell. And the reason these beads are binding is because all of these magnetic beads are functionalized to bind to the Ag protein, which is basically the constant region of the antibody right here. And so with all of these magnetic beads in our vial, we can very literally take a magnet and pull all of the beads to one side, and with them will come the cells that are bound. But if there's a cell that is not bound, it will be stuck in the supernatant, and we can extract that, remove that, and what we're left with is simply potentially disease-specific antibody-peptide interactions. So then once we then isolate these peptides and these antibodies, we then need to sequence that DNA, because it's the genetic information that we want. So we go through an elution process, we extract the DNA, we isolate the region within the plasmid that we want, because we only need a small portion of the plasmid. So once we've isolated that DNA like this, we then amplify it using PCR. And our goal is to basically increase our signal such that we do not miss a single antibody-peptide interaction. After we've amplified it, we send it to a sequencing lab, and we get back hundreds and hundreds upon thousands of sequences of DNA. Once at this stage, we're ready to put it into different software, different algorithms, and then convert this into data that we can use more easily. So the first software we use is called Immune, which was built in-house, and the idea of it is going to look at all of these sequences, and it's going to look for patterns. And once it finds patterns, it's going to start clustering them together. And as you can see, all of these have some stark similarities, and it's going to create what we call a motif. And a motif is a representation of a possible antigen that this antibody repertoire is binding to, that this antibody repertoire is specific to. And so we then want to take these motifs and look at them to see how enriched they are in the disease samples, and how either enriched or not enriched they are in healthy samples. And so we do that by then assessing the data. And so once we assess the data, we're going to divide here into groups of disease patients and groups of healthy patients, and we have their epitopes out like this. And this is all being done by a computer algorithm. And so the Immune will then look at these lists of epitopes and, again, look for similarities between the disease and the healthy patients, and it will then get rid of those, because those cannot be disease-specific if they're being found in both diseased and healthy patients. So what we're left with is basically what we've been working towards from the beginning, is potential candidates for disease-specific epitopes and antibodies. And so it is then these epitopes, these motifs, that we'll look up in a database called Hazard. So Hazard houses all of our antibody repertoires. And we can search motifs, much like the one we saw before, and find how enriched a sample is. So this motif could have been the one that we saw 2 slides back. And as you can see, it looks pretty enriched in this AMD disease samples and very much not enriched in the control samples, which is what we're looking for. Now, enrichment is a measurement of observed over expected. Observed is how many times did Immune sequence and read this motif as being a match for either this individual sample or this individual sample respective to which sample it's looking at. So how many times did they see that? And expected is statistically how many times should we have seen this interaction if every antibody interaction was truly random and didn't have some sort of specificity. And that's done based on a Poisson distribution and different statistical methods that we use in lab. So this is one example of I guess a motif chart. And this is something more common that we look at is, so here's our disease samples, and we want to compare possibly that same motif to a much wider range of samples. And as you can see, this motif is obviously not disease specific because A, not all of these people have AMD, and yeah, I mean that's basically it. These people don't have AMD, therefore they should not have an antibody reaction to something that could possibly be AMD specific. So this is a motif that can be eliminated. And this is a motif, something that we're looking for. And the reason this is a very interesting motif is because A, it shows high AMD specificity, or relatively high AMD specificity with low control enrichment. And then there's a much wider range of the population that is not showing a match for AMD. But this disease right here, these are Alzheimer's patients. And the reason this is interesting is because most Alzheimer's patients, as well as most people suffering from AMD, are elderly or are much older. And so something that we can look at is what percentage of the general population should be suffering from AMD, and does that match the percentage of people within this random grouping of Alzheimer's? And could that then show that these people just randomly have AMD based on statistical likelihood? So this is just a very interesting motif for Joel and myself, and something that we will look more into down the line. So that's kind of like how we look at motifs. That's how we look at whether something is disease specific. If we were to find an epitope that we could say, this is disease specific, this is something that is immunogenic to AMD, we would then want to work on finding the complementary antigen. And how we do that is by looking in protein databases. And so here's an example of a very well-known epitope, and this is the epitope for the human rhinovirus, or the common cold. And so this would be like the disease-specific epitope, or this would be the antigen that is immunogenic to the common cold. And we then want to be able to look this up in a database. We use one called BLAST, and then go forward and find the complementary protein, or the entire structure responsible for this disease. And so here's an example where this is very well known. This is fully charted. Here we have a capsid protein, or a capsid sheath, and it then relates to some bacterial cell for the rhinovirus. And so our hope is to do that with AMD, but we are simply not there yet. We don't have a candidate that could be disease specific, and even then, the protein database that we look into, they're not converging on any single protein. We're only really coming up with hypothetical proteins at this point, and it makes sense. We're not at a stage to be looking for a protein for AMD. And there's some acknowledgement, so I'd like to thank the Gorman program, specifically Kevin Gorman, Dean Pierre Woltzius, also the Dean. I'd also like to thank my lab, PI Patrick Doherty, my mentor Joel, and our fellow lab mate Michael Paul made the summer a great one, as well as the whole CSEP organization. They put a lot of time and effort into giving myself and many of my colleagues opportunities that just, frankly, many kids don't have. So thank you to people like Stephanie, Erica, Kelly, and everybody else in the CSEP program. Thank you. So if there are any questions, I now have time to answer.
Is there a way to, once you determine the disease-specific epitopes, is there a way will there be a way for you to test for it in advance? Like if I would like to know if that's going to happen to my eyes, right?
Mm-hmm. So that would be the idea, is if we find antibodies that are responsible for binding to something that is AMD, we could then theoretically can take an assay of like your blood and then, or just some biological sample, put these antibodies in that assay and then test for reactivity. And if there's reactivity in your sample, then it stands to reason that there's something in your body that is triggering AMD antibodies to react. AMD specific antibodies to react. And so that would be one way to reach a diagnosis or some kind of preemptive diagnosis.
So we're just about out of time for questions. I'm sorry about that. But we also have a lunch reception and we can follow up with these questions at the lunch reception.